For giving me the opportunity to present our work. I'm Olivier Henry, and I will represent the University of Rovira and Virgili in Tarragona, Spain, which is one of the members of the Mascot Consortium, which is a consortium that is consisted by 7 partners. You can see a range of names here and the organization underneath. And we set ourselves to develop the lab on a chip for rare tumor cell detection, involving breast cancer and in lung cancer. And I'll show you the results we have and how successful this project was. Okay. So just to give you a quick outline of what we wanted to develop. We wanted to develop a minimally invasive tool for the isolation and the characterization of circulating tumor cells in order to enable some therapy selection and monitoring. But that's also applicable to the detection of recurrent disease. You will see later that it's not wearable, it's not implantable, but it surely addresses the needs for personalized health. All those steps, the steps required for the isolation, characterization, and analysis of circulating tumor cells typically require a number of laboratory steps, which are very time-consuming, very tedious. And we start with a sample incubation, some cell isolation and detection. We then need to break the cells, the isolated tumor cells, to extract the genetic materials, to be further amplified and detected, and then integrate all this. And all those steps need to be integrated on a micro scale systems, using the lab on a chip concept. So what are circulating tumor cells, you will ask? They are basically shedded into circulation by primary tumors. So you have a tumor, in the case of breast cancer, breast tumor that will very slowly release circulating tumor cells, which may consequently cause some distant metastasis. And as an example, is for metastatic breast cancer, it's found that by detecting this level of circulating tumor cells or monitoring the evolution of the circulating tumor cells, you have a significant good prediction indicator for the overall survival of a patient. The detection is carried out in peripheral blood, usually, and assessed using different and is performed for different clinical applications. So as I mentioned before, you will want to do such analysis for the early diagnosis of a cancer and the case finding, or for prognosis and staging of a detected cancer, and then to monitor the therapy and see how the disease is being treated, or how fast is it coming back. So, really what it involves in terms of detections, this is a very challenging field. You will look at maybe 1 tumor cells in a milliliter of peripheral blood. And currently, this is achieved by, you have a number of techniques. Here are a few, but you have a density gradient centrifugation of the cells for the isolation of those cells, which are then immunostained and inspected microscopically. Or you might find some techniques that combine microscopy and some enrichment techniques. And there's a product called Cell Search by Veridex that do that. Or you can also use a combination of RT-PCR and, again, immunomagnetic separation, which is one of our partners' product, the ADNA test. But the challenge that is faced is to combine the immunomagnetic separations with a quantitative RT-PCR with multiple markers. And the next very challenging one that's been mentioned by speakers before me, that the integration is a crucial step and is a very challenging objective. So again, to take you through the steps of what's involved in a laboratory settings, you have your blood samples that we have. Maybe out of 5 cells, you have 1 tumor cells. The immunomagnetic separation means that we incubate the samples with some labeled magnetic beads. And by label, I mean that they have some, they are able to recognize some specific cancer marker at the surface of these tumor cells. So we then bind to it. And by simply applying a magnetic field, we can isolate this cell out of the healthy cells. That's the isolation separation. The cells then need to be lysed in order to extract the genetic material. Once extracted, we can remove, again, using taking advantages of the magnetisms of the beads. You can isolate the RNA, and by doing some RT-PCR and MLPA PCR, amplify the DNA present, and then detect, either microscopically or by electrophoresis. So you have seen that there are three distinctive steps to reach a conclusive results. You have the cell isolation, the mRNA extraction, and the DNA detection. That's what we set ourselves to do, to integrate those 3 modules with these 3 functions with, which will consist of different modules into a single instrument. So